Yes, so it's a pleasure to introduce Nishad Matange. Nishad is a faculty at IISER Pune, where his lab works on antimicrobial resistance in bacteria primarily, and that's the kind of work that I think he's going to talk to us about today. So welcome, Nishad, and we look forward to your talk. And everybody, please feel free to interrupt and unmute yourself and ask questions or raise your hand in between. Nishad is comfortable with that. So Nishad, take over, please.
Thanks, Deepa. I'm just going to go ahead and share my screen. Just give me a minute. Okay, so I hope things are visible. I guess they are. All right, good. Okay, so hi everyone. Really happy to be here. I wish I were giving this talk in person, but circumstances didn't really permit that, so here we are. All right, okay. So what I'm going to try to do today is give you a glimpse into the kind of work that my group has been interested in doing. I like to call this understanding the evolutionary logic of antimicrobial resistance. So let me take a minute or two to just explain what I mean. All right, okay. So all of us are familiar with natural selection. Either you've had rigorous training in evolution and you probably understand it better than me, or you have some functional idea of what natural selection is, how it works, or this workshop has given you that functional idea of how natural selection works. So essentially, we know that primarily, or a very large majority of evolutionary processes that we see around us can be explained by natural selection. Of course, there are instances where you cannot directly apply natural selection, but in a fairly large majority of cases, we know that this is true. And the idea of natural selection really rests on three main pillars. The first is, pre-existing variation. So in any population, we know that there is a natural variation in terms of how traits are expressed by different individuals in this population, and this variation also results in differential reproductive success. So we know that different variants have different propensities or probabilities of being represented in the next generation. Okay, and we know that phenotypes and phenotypic differences are heritable, which means that offsprings will resemble their parents rather than somebody else's parents. And all of this results finally in the selection of particular kinds of phenotypes over the other. And this, of course, depends heavily on the environment that natural selection is acting in, right? So the environment obviously plays a major role in this process. Okay, but as a molecular biologist, I'm interested in really trying to figure out what are the molecular underpinnings behind this, right? So can we figure out what each of these phenomenological ideas mean at the level of molecules and pathways and so on and so forth, right? So if you think about it, going from a variant to a selection of the variant in a population is really a process of integrating across multiple layers of organization, multiple levels of organization in biology. Right? For example, if you think about variants, these are typically, because we're talking about heritable variants, we're typically talking about genetic changes, things like mutations, right? So these mutations at the DNA level are going to result in mutant proteins. These proteins are going to have altered catalysis, let's say, or structural properties, so on and so forth. And these mutant proteins are then going to fit into some kind of a pathway, right? So you can think about metabolic pathways where multiple enzymes often act sequentially, right, to be able to produce some kind of an output. This pathway then integrates with other pathways in the cell, right, and finally gives rise to what we refer to as a phenotype at the level of the individual. But really, this isn't enough, right? Because selection isn't acting on this single individual; it's acting on this individual in the context of the phenotype of the other individuals in the population. So, if you think about it, you're really, talking about multiple levels of organization, and so understanding mechanisms means that you have to be able to make this journey. You have to be able to go from single mutations in DNA all the way to populations. And one of the best systems to be able to try and do this is antimicrobial resistance in bacteria, right? People have done this for decades now with a lot of success. And of course, the simplicity of the system, right, in terms of the ability to manipulate it, the ability to control variables, parameters in these kinds of experiments is a major advantage, right? In addition to the fact that bacteria are haploid, so what that means is that mutations are immediately expressed and so on and so forth, right? So antimicrobial resistance, of course, if we follow the natural selection logic, uh, uh, argues then one can argue then that in any population of bacteria, there are pre-existing mutants, right? These mutants have altered susceptibilities to antibiotics. And when presented with an antibiotic in the environment, these mutants will essentially get enriched, right? So much so that over extended periods of time, these mutants will take over the population. They will be fixed. The mutant alleles will be fixed in the population. And essentially, that's when we say that the population of bacteria have become drug resistant. Okay, so it's a very sort of straightforward framework that helps us understand a very fundamental process, and of course also has tremendous value because drug resistance in bacteria is sort of one of the major um, biomedical challenges that we're facing today. And in fact, it's predicted that after the COVID pandemic, we're probably going to be seeing very high numbers of drug-resistant isolates because most people are prophylactically being prescribed antibiotics during this period, right? So obviously a very, very relevant question to be asking. Okay, so in this regard, my group is essentially interested in asking three kinds or focusing on three aspects of the evolution of drug resistance in bacteria. The first of these is what has been referred to in literature as the fitness cost of drug resistance. So, through repeated observations over, again, several years, people have found, several tens of years, in fact, people have found that resistant bacteria tend to have lower fitnesses compared to sensitive bacteria in the absence of the drug. So what I mean is that if you were to take a resistant bacterium and you were to compete it with a drug-sensitive bacterium in the absence of the antibiotic, the drug-sensitive bacterium wins. And this is thought to be due to the fact that resistance conferring mutations usually occur in fairly quote unquote important genes of bacteria, right? So housekeeping genes, for example, you can think about the ribosome, you can think about the RNA polymerase. These are all pathways that antibiotics tend to target, right? And therefore mutations that confer resistance tend to occur in these proteins, therefore, altering the catalytic activities of these very important enzymes in bacterial cells, right? And this obviously reduces growth rates and therefore results in a fitness cost, right? So we're interested in asking, where do these fitness costs come from? Do all of them come from changes in catalytic activities? What is the contribution of structural perturbation, for example, to fitness cost, and so on and so forth? So those are the kind of things that we do. The second sort of area that we like to think a lot about is the impact of different drug concentrations or exposures during the evolution of antimicrobial resistance. And I'm going to focus a little bit on this for the rest of the talk today. And finally, I'm trained as a biochemist and a geneticist, so my pet passion is to kind of try and figure out, how the genetics of antimicrobial resistance relate to the environments that they're selected in. So what I mean by this is that let's say you have 4 or 5 different kinds of mutations that can potentially confer resistance. How does the environment, or how do changes in the environment select for one or more of these multiple options, if you will, that are available to bacterial species, right? And so today's talk is going to focus largely on the second and third aspects of the focus of the lab. So, so I mentioned in passing about drug concentrations, and our interest in trying to understand how different drug concentrations can select for antimicrobial resistance. The kind of underlying motivation for this, of course, comes from the fact that bacteria experience antibiotics in a number of different kinds of environments. And turns out that in different environments, the concentrations of drugs can span over orders of magnitude. So if you think about bacteria in your body that are experiencing antibiotic, which is being consumed by you as a therapeutic, right? These antibiotics are typically given at very high concentrations to be able to eliminate, all of the pathogen from your body. And so what they're experiencing are lethal concentrations of the antibiotic. But bacteria are, of course, facing antibiotics not just in your body, they're also facing them in soil, in, water bodies, in all sort of natural reservoirs in which bacteria live, right? And how do antibiotics get into these places? Well, they could be derived from natural sources. For example, think of fungi, think of other bacteria that produce antimicrobials that live naturally in soil. or of course also coming from human activities. So we use antibiotics in a variety of different spheres, right? So apart from, of course, as therapeutics for humans, we use antibiotics routinely in animal husbandry, right? They're used as growth promoters. They're used as prophylactics, right? And so in all of these cases, runoffs from these sources can essentially end up in soil, in water, and um, inadvertently you end up exposing bacteria to antibiotics. The other source of antibiotics, of course, major source of antibiotics is chemical plants that produce antibiotics, and effluents, are typically let out into water bodies, and the antibiotic gets into the environment. So what's important to note is that in all of these natural environments, the drug concentration, because the drug gets diluted out, right? So the drug concentrations that bacteria are likely to face are extremely low. In fact, these are usually sublethal concentrations. But there's mounting evidence now to suggest that these could, in fact, these environments where you have sublethal drug concentrations could actually be facilitators of antimicrobial resistance. And so so my lab is really quite interested in understanding, how resistance evolves at sublethal versus lethal concentrations of antibiotic. Okay. In this regard, over the past 10 or so years, there has been some amount of work that has come out from my group and a lot of other groups around the world that have shown that evolutionary trajectories, evolutionary genetic trajectories to resistance are very often different in low and high drug pressure environments, right? And the story that I want to tell you today is related essentially to this idea that the genetics of adaptation to high and low drug pressure environments can often be very different, right? And it produces really interesting um, uh, mix of genotypes that are worth keeping in mind when you think about how bacteria adapt to antibiotics, right? Okay, the other thing that you need to know before I get into the meat of the matter is that adaptation to antibiotics is actually not restricted to resistance, right? So resistance is one of the most popular ways in which we know of bacteria being able to tackle antibiotics. But in fact, adaptation to antibiotics comes in a number of flavors, right? So of course you have classic sort of resistance, which is defined by a very clinical phenotype, right? The inability to kill a bacterium or treat an infection with an antibiotic. But we know that bacteria evolve other kinds of phenotypes. For example, tolerance and persistence. Some of you may have come across this in literature, and if you feel like, we can talk about this at the end of my talk. And what we're starting to realize is that these phenotypes could be precursors to resistance. So these could be sort of gateways to allowing bacteria to evolve resistance, and that's something that is emerging now as an area of interest for people interested in this kind of thing. Focused on this kind of thing. The other important thing, of course, is also that antibiotics, particularly at sublethal doses, can induce phenotypic changes in bacteria, right? That can change susceptibilities. And so this is, remember, happening without any genetic change. It's just that in response to the antibiotic stress, bacteria change things like their colonial behavior, right? So essentially, they start forming pellicles, they start forming biofilms, they upregulate a number of drug efflux pumps, and all of these things are going to finally affect how susceptible a bacterial population is to an antibiotic. Okay, so what we want to do, sort of the holy grail for me, is to be able to take strength of antibiotic or strength of selection that the bacteria are facing, combine it with how different modes of adaptation are used by bacteria to survive in the face of antibiotic challenge, and to be able to explain why certain modes of adaptation, certain mechanisms of resistance work under certain strengths of selection at the molecular level, right? And that's really, for me, the way I'm thinking about this problem. Okay, so I'm going to tell you a story today that was published late last year, and this essentially tries to understand this issue of how different strengths of selection can select for different kinds of strategies of adaptation in bacteria, and whether we can find molecular explanations for this. So the system that we are going to look at is trimethoprim resistance in E. coli. Let me just introduce what this is. So trimethoprim is an antibiotic that is used along with sulfamethoxazole as a treatment for gram-negative bacterial infections. very often used for UTIs, for urinary tract infections caused by Escherichia coli, right? Trimethoprim is a synthetic antibiotic. It's an antifolate, very similar to methotrexate, which is used for mammalian cells, right, for cancers and things like that. And it inhibits an enzyme called dihydrofolate reductase, DHFR for short, and I'll be calling it DHFR for the rest of the talk. DHFR is an enzyme that's involved in the synthesis of tetrahydrofolate, right? And tetrahydrofolate is a fairly important cofactor, which is used for the biosynthesis of pyrimidines and amino acids. So you can imagine now that cells treated with bacterial cells treated with trimethoprim will not have any active DHFR. Therefore, there won't be tetrahydrofolate produced, which will shut down pyrimidine biosynthesis, which will shut down DNA synthesis, and this will eventually lead to stasis of growth, right? So trimethoprim is a bacteriostatic antibiotic. So we know a couple of mechanisms by which E. coli can evolve resistance to trimethoprim. One of the mechanisms involves horizontally acquired DHFR enzymes that are trimethoprim resistant. So these are typically these spread through populations via integrons or plasmids, right? This is a fairly important clinical source of resistance, but we're not going to talk about this today. What we're going to focus on is chromosomal mutations which occur in the DHFR enzyme itself. So the enzyme has a trimethoprim binding site, and mutations typically map to the binding site for trimethoprim, which is again fairly intuitive, essentially preventing trimethoprim binding and therefore allowing the cell to resist trimethoprim, right? So in addition to these resistance mechanisms, we recently discovered another way by which Escherichia coli can adapt when confronted with trimethoprim. This really, came up fairly serendipitously when we were sequencing out the genomes of some trimethoprim-resistant bacteria from the lab. And we found that pretty much in all isolates that we sequenced, we found loss of function mutations at a locus called MGRB. Okay, and I want to spend a minute talking to you about this because MGRB and DHFR are two of the main players that I'm going to talk to you about in the subsequent slides. So MGRB mutations typically involved either frameshift mutations in the coding sequence or insertion of transposable elements, IS elements, into the promoter of this gene, and essentially turned this gene off, right? So all of the mutations that we found in MGRB were loss of function mutations. So remember, these were mutations that we found in trimethoprim resistant bacteria. Right. And so in order to see if MGRB changes susceptibilities to trimethoprim, what we did was we generated a knockout of MGRB in E. coli and looked for how its susceptibility changed to the antibiotic and compared it with wild type. Now, the knockout of MGRB shows a very interesting phenotype, which is that, so the gray trace here is the knockout for MGRB, and the black trace in this graph is the trace for wild type. And we're essentially looking at growth of these bacteria as a function of trimethoprim concentration. And what you will see is that the minimum concentration needed to inhibit the growth of E. coli was the same, whether we looked at wild type or at the delta MGRB strain. However, at sublethal concentrations of the antibiotic or at sub-MIC concentrations of the antibiotic, the mutant actually does better. And so this is a classic drug-tolerant phenotype where you don't really get very good growth of the mutant at high concentrations of the antibiotic, but you get better growth at lower concentrations, right? We did a series of genetic and biochemical experiments to try and understand why, and it turns out that the reason for this is really related to how MGRB affects the expression levels of DHFR in a cell. So we know that MGRB is a very small protein. It's a membrane protein, which is part of a very popular and very well characterized signaling pathway in gram-negative bacteria, which is called the 4PQ signaling pathway. So this is a classic two-component system pathway for those of you who are familiar with this sort of signaling. If you're not, it doesn't matter. Essentially, 4Q is a membrane-bound receptor which gets activated by extracellular cues. Right? It then phosphorylates 4P, which is a cytosolic protein. Phospho-4 protein is a transcription factor itself, which can go and activate the transcription of a number of genes, which involve, stress response factors, other transcription factors, which can, change transcriptional profiles of the cell. And it turns out that one of the targets of 4P is in fact the DHFR enzyme itself, right? And so, so essentially, DHFR will be the transcription of DHFR will be activated when 4P is active, which will result in higher amounts of DHFR, which will, therefore, if you increase the drug target in a cell, you need greater amounts of antibiotic to achieve the same level of inhibition. And where MGRB fits into this picture is that MGRB is also a target of FOP, right? But it's in fact a negative feedback loop, right? So MGRB, when it's made, goes and binds to FOQ and shuts this signaling off. So it's sort of like a brake on the system, right? And so when you have a loss of function mutation in MGRB, you remove the brake, right? So you hyperactivate the signaling, which leads to larger amounts of DHFR being produced by the cell, which leads to trimethoprim-resistance, right? And so we elucidated this mechanism, which added to the list of different kinds of ways in which E. coli can tackle this antibiotic, right? So we've spoken about resistance mechanisms, and we've spoken about tolerance mechanisms. And for the rest of the talk, I need you to remember chromosomal mutations in DHFR, which confer trimethoprim resistance, loss of function mutations in MGRB, which hyperactivate 4PQ, which lead to transcriptional upregulation of DHFR and lead to trimethoprim tolerance, right? These are going to be the two main players for the rest of the talk. Okay, all right. So we were interested, as I said, in understanding how adaptation occurs at different selection pressures. And so the way we decided to answer this question is by using a laboratory evolution approach. And essentially, we were going to evolve, or we plan to evolve wild type E. coli, drug sensitive E. coli at two different drug pressures, low and high, right, for some number of generations, and essentially look at what happens, right? So fairly straightforward experimental setup. In order to select concentrations of trimethoprim that corresponded to low and high pressures, we did a standard, dose response kind of experiment where we grew drug sensitive wild type E. coli in different concentrations of trimethoprim, and using this the trend of growth that we observed, we selected a low and a high concentration of trimethoprim, which I'm going to essentially just call low and high, right? So the specific concentrations are on the slide, right? What we then do is that we had three replicate lineages evolving in each of these two drug pressures. So we had a total of 6 evolving lineages, 3 experiencing low trimethoprim exposure or low trimethoprim pressure, and 3 experiencing high trimethoprim pressure. And we did this with a standard sort of serial passage protocol for about 350 generations. So these bacteria are experiencing these concentrations of trimethoprim for approximately 350 generations. Okay, so this was the evolution experiment that we set up.
And the question?
Yes, please do.
Yeah, so on the left side, this growth is shown at the end of all the 350 generations or at some intermediate time point?
No, so the growth that I'm showing you is just for the drug sensitive ancestor of the evolution experiment. At the end of it, of course, I'll come to what happens at the end of the 350 generations, right? So the graph here is just for the ancestral populations.
So it's not exposed to ancestor means for the wild type. Is that what you mean?
Right.
Okay, fine.
Right, so this is a naive population. They haven't seen trimethoprim before this. Shall I go ahead?
Yes, please. Thank you.
All right, Kavita. Okay, so the first thing that we wanted to ask was that, does the selection of resistance in these two different kinds of at these two different drug pressures, does it really change, right? Is it really different? So there's a very simple way for us to do this. Essentially, what we do is that we had our high pressure lineages and our low pressure lineages, right? And at different time points, we plate them out on, obviously we plate them out quantitatively on agar plates which contain high concentrations of trimethoprim, such that only those bacteria that have evolved resistance will form colonies. And parallelly, we plate them out on drug-free media where you will get the total population size essentially, right? And what you find is plotted graphically on the right here. So what I'm showing you is the titer of resistant bacteria, right? And the carrying capacity is essentially telling you that the total population size isn't really changing through the evolution experiment. Okay, the graph on top is telling you what's happening at high drug pressures, and the graph below it is showing you what's happening at low drug pressure. So the first thing I need you to appreciate is that, of course, there is some difference between the replicates that we're seeing at a particular drug pressure. But what's important is at the end of 350 generations of evolution, right, resistant bacteria have basically got fixed in the high in the high drug pressure lineages, right? More than 99% of the bacteria in these populations are drug resistant, right? On the other hand, when you allow these bacteria to evolve resistance at low drug pressures, the numbers remain extremely low, right? And in fact, in one of the lineages, the numbers don't increase at all over these 350 generations, right? Resistant bacteria typically are less than 1% of the population, right?
Nishad,
And yeah,
clarification.
sure.
Is the low pressure population also tested at high drug dosage, or is it
Yes.
at the dosage at which it evolved?
No, at high drug dosage.
So
So we're
both
essentially
shots.
looking, yeah. So essentially, we're looking for bacteria that have a higher MIC than the wild type.
Okay, great. Thanks.
All right. So we, so one idea here was that, okay, so maybe the low pressure lineages didn't have enough time to fix resistance. So for one of these lineages, we continued our evolution experiment beyond 350 generations, and we went all the way to about 500 generations, and we really didn't see much of a change in the proportion of bacteria in the population that were drug resistant, right? So what this told us then is that resistance evolves at both drug pressures, right, at high and low drug pressures. But the frequency in the population of resistant bacteria seems to be affected by the pressure that of the drug that we had imposed on these populations, right? Now, at the face of it, this seems really intuitive, right? That at low drug pressures, you get lower titers of drug-resistant bacteria, but we decided to dig a little bit deeper to see if something more interesting was happening here. So the way that we chose to do this was using genome sequencing. So what we did was that from extremely early time points, right? So something like 20 generations of evolution, at which point we really have almost undetectable numbers of resistant bacteria. We used genome sequencing to figure out what were the changes that had occurred in these populations. So the entire population was essentially subjected to NGS, and we were looking specifically for two things. The first is we were looking for mutations in the DHFR enzyme. Remember, DHFR mutations we know confer trimethoprim-resistance, and since in line with the fact that we weren't really detecting any trimethoprim-resistant bacteria, we found that there were no real mutations that we could identify in the DHFR enzyme itself. What was interesting, though, was that the MGRB mutations, remember, these are mutations that lead to loss of MGRB function and confer trimethoprim tolerance, right? These mutations had already swept through the populations, right? So whether we look at populations that came from high selection pressures or low selection pressures, roughly 70 to 80% of bacteria in these populations had already accumulated loss of function mutations in MGRB. So what this told us was that irrespective of what the selection pressure is, tolerance seems to precede resistance, right? And occurs fairly rapidly, right? Within 20 generations of selection, we've had this massive sweep of MGRB mutations in the population. So what happens if we now follow these resistant bacteria through to later time points, right? So what we did then was that we isolated out specifically resistant bacteria from the high and the low drug pressure regimes and subjected them to genome sequencing. And again, we're looking essentially for mutations in DHFR and in MGRB, and of course, anything else that seems interesting, right? So here, in line with the fact that we have specifically sequenced resistant clones from these populations, all of these bacteria had mutations in DHFR. You would expect this, right? But what was interesting was that all resistant bacteria also had mutations in MGRB, right? So what this told us was that resistance was evolving in a tolerant background, right? So tolerance preceded resistance, and then in a drug-tolerant background, additional mutations that conferred resistance were getting fixed in the population, and these were mutations in DHFR, right? What was also interesting was that actually there weren't really any detectable mutations that were different between resistant bacteria that were derived from high pressure and low pressure lineages, right? So what this told us was that resistance evolved at both drug pressures. It evolved from tolerant intermediates, right? And even though their titers were extremely different in the two kinds of lineages that we had, the mutations that you get were pretty much the same, right? So what we were seeing was something that looked like this. Sure.
Sorry, I missed this then. So then what is the difference genetically between the tolerant and the resistant? They have the same mutations, right? So you said that tolerant ones came first and then they became resistant, but genetically the same, or?
No. So the tolerant ones have mutations only in MGRB, right?
Yeah.
Which leads to upregulation of DHFR levels, right? The resistant ones have mutations in MGRB and they have mutations in DHFR itself, which prevent drug binding.
Okay, I see.
Right? But it doesn't matter whether you get them from low or high selection pressures. They usually have this, or they have the same mutations in both cases.
So genetically, okay, they are different, the tolerant and the resistant one, and but however they are same whether you have a low selection and high selection.
Precisely, yeah.
But the numbers are low, although they are genetically similar in both selection pressures. Is that what you're saying?
Precisely, yes.
Okay, do you know? Are you going to explain that? Why
Yes.
doesn't? Oh, okay.
Yes. And I think I have a reasonable explanation for that. So we'll come to that in a bit. All right, good. So, so like Kavita very nicely summarized, actually. So we were looking at a couple of phenotypic transitions that were occurring, in our evolving lineages, right? Obviously, all lineages started off as drug sensitive, right? They first transitioned from drug sensitive to drug tolerant, okay? And this was mediated by mutations at a particular locus, which is the MGRB locus, right? This happened for a majority of the population, irrespective of whether they were seeing high drug concentrations or low drug concentrations, right? What was interesting, though, was that there was a second phenotypic transition that occurred, which took drug-tolerant bacteria and converted them into drug-resistant bacteria, right? And this was mediated by mutations in DHFR. The FOLA is the gene that codes for the DHFR enzyme. And here's where the difference really was dramatic, which was that a majority of bacteria in the high drug pressure lineages made the transition from tolerant to resistant, right? But only a minority of the bacteria did this when selection pressures were low, right? So what this prompted us to ask, like Kavita very nicely pointed out, was what was resulting in this difference, right? Why were these tolerant populations, which were roughly at the same frequency in the population as in the high drug pressure lines, why weren't these transitioning as much to resistance, right? And were there other evolutionary trajectories that these populations took? Okay, so in order to do this, we went back to some genome sequencing, and what we did was that from our low drug pressure lineages, we essentially separated out drug-resistant bacteria and drug-tolerant bacteria from different time points, right? So at different numbers of generations of evolution, we got drug-resistant isolates and drug-tolerant isolates, right? And we subjected them to genome sequencing. And here's where some really rather puzzling results came up. So of course, like I said, initially you only had MGRB mutations, which confer drug tolerance, right? A minority of the bacteria fix mutations in FOLA or DHFR in addition to MGRB. This converts them to drug resistant. But what we found was that there was another lineage of evolution that we could trace, right? Another genetic lineage of evolution that we could trace in the same population, right? Where drug tolerant bacteria fixed mutations in another gene, right? And this gene is called RpoS, right? And this occurred in 100% of tolerant bacteria that we sequenced from later time points. So this mutation was occurring in the background of MGRB and was occurring, almost, across the board in all of these tolerant bacteria, right? What was interesting was that these mutations were again loss of function mutations in RpoS, and I'm going to tell you in a bit what RpoS is. So we had generated a knockout of RpoS to see if loss of RpoS itself confers some kind of advantage in trimethoprim. Turns out it doesn't, right? So so this was puzzling because you had this mutation that was again sweeping through the population, right? But by itself, the mutation doesn't really confer much of an advantage to trimethoprim. Yeah, Deepa, go ahead.
Actually, I may have missed this, but
Sure.
how do you know this mutation is sweeping through the population? You've isolated tolerant mutants, and then sort of in that population, subpopulation, it seems to be fixed.
Sure. Okay.
What fraction of the total population has the RpoS mutation?
Has RpoS, right? So the way that we isolate the tolerant bacteria is that actually we don't do anything special. We just sort of streak out these strains onto non-selective media, which means just plain LA, right? And we randomly pick colonies, check for their MICs. If their MICs are the same as wild type, but they have MGRB mutations. We call them tolerant, right? And so we did this for about 10 or 20 clones, which were randomly picked, and they all had RpoS mutations. So I don't really know that the mutation frequency was 100%, but given the way that we were doing it, which was fairly non-directed, I would suspect that most bacteria in these populations have the RpoS mutation.
Okay, makes sense. Thanks. But you don't have, for example, the population level sequencing that you did for the early time point.
No, we didn't. We didn't do it at this time point. But I have some other circumstantial evidence to suggest that this is happening in a majority of cells. So RpoS has loss of RpoS activity has a very easy to measure functional readout, which is sort of like a colonies are red or green kind of a readout. And if you just spot the population on these plates, the population phenotype is a loss of function RpoS phenotype. So it looks like a majority of the cells don't have RpoS.
Okay, thanks. Yeah.
Okay, so this sort of puzzled us, and we wanted to know what RpoS was doing in all of this, right? So let me tell you a little bit about this gene, right? So RpoS, for those of you who are bacteriologists, will of course know this gene rather well. It's a sigma factor, right? So essentially, it codes for a protein which forms part of the RNA polymerase holoenzyme, right? And sigma factors will determine which promoters the RNA polymerase gets recruited to. So they will decide what genes are transcribed in any bacterial cell. And what's interesting is that RpoS competes with another sigma factor called RpoD in a bacterial cell, right? RpoD activates genes involved in growth and division, right? Growth and reproduction. Whereas RpoS activates genes involved in stress response, right? So you have these two sigma factors that are now competing with each other for occupancy on the RNA polymerase, right? And the one that sort of is able to occupy the RNA polymerase will determine whether the bacterium starts to, kickstarts stress response pathways or growth and division pathways, right? What's even more interesting for us is that RpoS is itself a target of FOP. Right? Remember, FOPQ is the signaling pathway that was activated because of loss of the MGRB gene, right? So what we're expecting is that these cells, which have the drug-tolerant cells, right, which have lost MGRB, essentially have hyperactivated FOPQ, and therefore they're producing tons of RpoS, okay? And when you do this, you're going to bias the transcriptome of these bacteria in favor of stress response genes and away from genes involved in growth and reproduction. And this in itself could be a source of a fitness cost for the cell. Right? And so we argued that maybe RpoS mutations actually act as cost compensation for the drug-tolerant phenotype that we're seeing, right? But in order to kind of verify this, we had to make sure that there is a cost to drug tolerance, right? So the way we do this is in two different ways. The first is we, of course, measure the relative fitness of a delta MGRB strain and find relative to the wild type, right? And in the absence of the antibiotic, we find that there's about a 20% lower relative fitness for the delta MGRB strain, right? Is this coming because of hyperactive FOPQ leading to greater levels of RpoS, leading to stress response genes being transcribed at the expense of growth genes? Well, one way to do this is to pharmacologically turn this signaling off. So if we were to treat a delta MGRB strain with an inhibitor of FOQ, then we should be able to relieve some of this fitness cost. And we can do this with something as simple as magnesium ions, which we know bind to FOQ and sort of shut its activity down. And the red bar essentially is relative fitness of the delta MGRB strain in the presence of magnesium. So obviously you see that at least some of the fitness is gained. Right? We can also do this genetically by simply knocking out FOQ or FOP. And in both of these cases, what you find is that some of the fitness cost of loss of MGRB and hyperactivation of FOPQ is in fact alleviated, right? Yes, Deepa.
Sorry, I keep having, there's a lot going on here, so I might be asking daft questions, but are these assays in the presence of antibiotic or absence of antibiotic?
These are in the absence of antibiotic.
Okay, so you're saying there's a fitness cost of overexpressing FOQ in the absence of antibiotic, and then maybe that explains why we see whatever we see.
Precisely.
Okay, thanks.
Yeah. All right. Good. So essentially, this helped us to establish that drug tolerance is also costly, right? And the overactivation of FOP4Q perhaps leads to the overactivation of RpoS, which is what is giving us this fitness cost, right? So if we were then to if we wanted to experimentally test this, how would we do it? Well, the way we chose to do it was again by using strain competitions, right? So we generated RpoS mutants, right? Remember that our sequencing had told us that all mutations were loss of function mutations in RpoS. So we just went ahead and generated a delta RpoS strain, a knockout for RpoS. And then what we do is that we mix the RpoS mutant with wild type, or we mix an RpoS MGRB double mutant, right, with the MGRB strain, right, the MGRB knockout strain. And what we do is that we allow these combinations to compete with one another, either in the absence of the drug, right, or at low or at high trimethoprim pressures, right, which is what we've used for our evolution experiments. And essentially what we're doing is that we're looking for change in ratios, right? So the initial ratio, if it gets maintained after the competition, we know that RpoS really isn't contributing to the fitness of these strains. But if the ratios change, then we know that RpoS changes the fitness of these strains. And what you see from the graph on the right is, so our initial mixing ratio is 99:1. The RpoS knockout strain is always in minority, right? When you look at, so the black graphs are essentially a delta RpoS strain being generated in a wild type background. So this strain has normal levels of 4P, 4Q activity, right? And RpoS deletion in this strain essentially, is actually costly, right? Irrespective of whether you're in no drug or in low or high drug, right? The ratios essentially get biased to, in favor of the wild type, right? But the same RpoS mutation generated in a delta MGRB background confers an advantage. And this advantage is again independent of whether or not trimethoprim is present in the medium. So the reason we think that RpoS was getting favored as the mutation in these low drug pressure lineages was because it was simply compensating for the costs of MGRB mutation, right? But this still didn't really tell us why low drug pressure lineages went down this route of adaptation, right? Which means they got up to drug tolerance, but then majority of the population went towards cost compensation rather than drug resistance, whereas the high pressure lineages went took the resistance route, right? And one possible explanation for this, we thought, could be that resistance is a fitness maxima only when you have enough drug present in the environment, right? So unless there's sufficient drug to kind of manifest the benefit of a resistance conferring mutation, maybe it's better to compensate for your costs rather than gain a new mutation in a resistance conferring gene. And so in order to be able to test this, we once again decided that we will use strain competitions, right? And this time, instead of competing 2 strains, we decided that we'll just have a little bit of fun with it and we'll compete 3 strains. Okay, so what we did was that we mixed tolerant bacteria. Remember, these bacteria have mutations only in MGRB, right? We mix them with resistant bacteria, which have mutations in MGRB as well as DHFR, right? Or cost-free tolerant, right? Cost-compensated tolerant bacteria, which have mutations in MGRB and RpoS, right? And remember that tolerant bacteria will either convert to resistant or to cost compensated tolerant depending on the kind of drug pressures that they were exposed to, right? So we mix all 3 bacteria together. We keep the resistant and the cost compensated tolerant in minority, and we allow these 3 strains now to compete with each other at different trimethoprim concentrations, and once again look at changes in ratios of the genotypes, right? And what we find is, in fact, exactly what we found from our evolution experiments, which is that so the first bar is the initial fraction of the population that was tolerant versus resistant versus cost compensated tolerant, right? And because we biased it in favor of the tolerant bacteria, you see a massive black bar here and just a sliver of a band for the red and the orange, which are resistant and the cost compensated tolerant, right? And now when we look at these low antibiotic concentrations, turns out that the cost compensated tolerant was in fact enriched compared to the others, right? But as you start moving up the gradient of antibiotic, you find that resistance becomes the favored strategy. And so we were able to directly show that the strategy for adaptation, right, that is most successful is dependent very heavily on the drug pressures that they're experiencing, right? So cost compensation is favored at low drug concentrations and resistance is favored at high concentrations. So this allowed us to kind of take molecular players and fit them into why certain kinds of phenotypes were getting selected under certain kinds of pressures versus at a different kind of pressure, right? And it all kind of bottled down to an evolutionary branch point that we find tolerant bacteria have access to, right? So they can either compensate for costs or they can go the resistance path, and depending on what your selection strength is, which path these bacteria will take will be determined, right? Okay, so that's all the data that I have for you today. What I want to leave you with is, so if you've basically lost me through the talk, here's where you probably want to listen. So here's what I want you to take home from this talk, right? The first important point is that there are many ways to improve fitness in an environment, right? When we talk about phenotypes like drug resistance, we tend to forget this, right? And we tend to focus on phenotypes that are either most obvious or most well characterized. But in fact, what you find is that most organisms, bacteria included, will have many tricks up their sleeve, right? Many ways to enhance fitness in any given environment, right? And the strength of selection seems to be an important factor that determines which of these different adaptive strategies are going to be the most successful, right? So the fitness maxima of a particular kind of will be determined by the kind of environment that you're working in. And in our case, all we've done is titrated the amount of antibiotics. So it's really the same environmental factor, but just at different concentrations, and you can end up producing or you can end up biasing these trajectories towards one or the other path, right? What's also important to remember is that adaptive strategies are not insulated from one another. So remember, I told you that cost compensation through RpoS mutations is only valid when you have evolved tolerance, right? In a wild type background, it does nothing. And so this kind of epistasis is something that you have to keep in mind when you're looking at how adaptation is occurring and what strains get selected and why, right? And finally, of course, one can look for molecular mechanistic explanations for the genetic structures of adapting populations. And I think that this is something that really excites me, and I hope that I'm able to convey that excitement to you. Yeah, that's all I have for you today, and I'm happy to take questions.
Very nice talk. Thanks, Nishad. People have questions? Subrata?
Yeah.
And that was really interesting, Nishad. But I have a couple of questions.
Sure.
So, yeah, did you try a within subject design, like for the in the low drug concentrations? Did you again try and put the same lines to a high drug concentration as well to see, if you do get resistant mutants there?
Right. No, we didn't do that. That's a great idea. We didn't do it for this study. We did it for another study that looked at something similar, but we haven't done it here. No.
Okay. And I was wondering, could you help me just know the difference between tolerance and persistence? It was always confusing. Yeah.
Sure. Okay. All right. So tolerance is a population. So tolerance and persistence both are population level phenotypes, right? Essentially, persistence, however, is where a subpopulation of bacteria will survive in face of antibiotic insult, right? And persistence is thought to come because of the natural variation in growth rates that bacteria exhibit, right? So something like 1% of a bacterial population naturally has a lower growth rate than the mean, right? And because it has a lower growth rate, antibiotics are less effective against it. So when you have antibiotic present, these kind of sluggish bacteria will just hang around, okay? But the numbers will be typically very small. It will be 1% or lower. I think it may be something like 0.1% or lower, right? So that's persistence. Tolerance is usually where the entire population, so it's not occurring in a subpopulation, it's occurring in a majority of the population. But here what's happening is that you're really dying slower than the wild type is dying, right? And this is very often genetically triggered.
Oh, okay.
Does that help?
Yeah. And I have another question. Sure. So you spoke about mutations, chromosomal mutations that you're looking at currently, and then you also mentioned horizontal gene transfer. So what are the difference in the rates? Like, which are actually more common, more often found? Like, is it by HGT or is it?
So it depends on where you look. There are some, so for example, gram-negative bacteria, things like E. coli, Salmonella, etc., very often are biased towards horizontal gene transfer. Okay, bacteria like mycobacteria have almost no horizontal gene transfer, so their mutations are primarily chromosomal.
No, I see. Okay.
So the answer depends quite a bit on the species that you're looking at.
I see. Okay. Thank you. Thank you. Yeah.
Maybe I'll ask a question. So could you go back to this very early slide you had shown where you had delineated the MIC of both your MGRB mutant and the wild type?
Was it this?
Maybe, well, no. One before that where you had shown where 4P upregulate. Yeah, this one.
Yeah.
So on this, could you point out the two concentrations of trimethoprim that you use for the low and the high?
Sure. So, can you see my
Yes.
hand? Yeah. Okay. So the low concentration is 50 nanograms per ml. It's somewhere here. This would be where 50 nanograms per ml is, and 300 nanograms per ml is the high concentration, which is somewhere here.
Okay, maybe that explains. So what I was wondering is, in the case of high selection pressure, why do you get MGRB mutations in the first place, right?
Yeah.
And maybe the slight advantage is what allows that to happen.
Yeah.
Okay, so
actually, the other thing, Deepa, is also that most of these MGRB mutations are occurring they're occurring because of IS element insertions. So just the frequency of these mutations is also going to be much higher than everything else in the cell.
Yeah.
So that's probably also biasing it, in that direction.
Yeah, mutation bias. Yes. Yeah. Sounds like yeah. No, I was just, because I was curious, so okay. So then you'd expect them that if you evolve lineages at a super high concentration of trimethoprim, then this MGRB is just not an option anymore.
Irrelevant. Yeah, this is what we're thinking. Yeah. Problem with evolving them at extremely high concentrations is that you only end up selecting for pre-existing mutants because the population just crashes, right? So those experiments we haven't really been able to convincingly do.
Right. Right, makes sense. And I guess then what Subrata was suggesting, where you sort of gradually increase the concentration, would be another way to think of how the population converts. Yeah, this is very interesting. Thanks. Any other questions? Anybody else?
I had a little question.
Go ahead.
Sure.
In the 3-strain competition that you showed,
right?
Are there more bars? Are there like more concentrations at which you've tested?
Yes. Do you want me to go to those data? Is this Bruhat?
Yes, I am. Hi.
Okay, Bruhat. Hi. Okay. Do you want me to go to the graph or?
No, that's fine. I mean, I was just wondering at what point the resistant ones.
Yeah. So actually, the idea there was to be able to identify that breakpoint concentration that will essentially tell us, whether you go towards resistant or towards cost compensated, right? There was no clear answer. It was sort of a very gradual trend, and also those experiments, weren't quantitatively as reproducible as we would have liked them to be. So, out of a couple of times where you do the experiment at the extremes, you see more or less the same thing, but in the middle, you kind of, it doesn't really work that well. Sure. So I don't really have a good answer for you, to be honest.
Okay. Yeah. Thanks.
I have one more sort of maybe unfair question, but I'm curious. I imagine there are many ways to upregulate 4Q, right? And I'm surprised that only this one sort of pops up again and again.
Yeah.
Do you have any intuition based on the pathway or something, why this is the case?
Okay, so in a very rare subset of isolates that we sequenced, we found point mutations in 4P itself. Okay, but they were so rare that we didn't know how seriously to take them. Again, I think it's going to boil down to the IS element versus point mutation bias, right? Which is just, it's a massive bias, right? IS elements just move around like crazy, and so that's probably what's giving us this.
Yeah, no, that makes sense. Yeah. All right, if there are no more questions, thank you very much, Nishad, for an exciting talk, and good luck with the next experiments, and hope to hear from you about those soon.
Thanks.
Thank you, Nishad. This is a nice talk. I enjoyed it. Thank you.
Thanks, Kavita. Thanks.
Bye.
Bye bye. See you.